Hello everyone, and thank you for being here. I'm honored to kick off our talks, and as the introductory speaker, I'd like to take a short moment to acknowledge the extraordinary work that Julia and Mr. Shapiro and everyone at UCSF are doing with this program. This opportunity has had a profound effect upon my own education and interest in science, and I can confidently say that it has done the same for all of my peers. And so to Dr. Kevin Shannon as a previous director of the MSTP program, and to Dr. Mark Anderson as the current director, and to all PIs who were so generous as to take Lowell students this summer, I would like to give a big thank you from myself, from my peers, and from future Lowell students for having faith in us. So without further ado, what I'm here to talk about today is my summer research project, restoring GTPase activating protein activity to NF1 deficient acute myeloid leukemia. So first, for some background, shown here is a diagram of the RAS pathway. And RAS is a protein active in near the membrane of cells that, when phosphorylated, begins a signaling cascade that results in cell growth, proliferation, and survival. And RAS is active when bound to a guanosine triphosphate, or GTP, and inactive when bound to a guanosine diphosphate, or GDP. And NF1 is a GTPase activating protein, or GAP, meaning that it binds to RAS and accelerates the hydrolysis of GTP to GDP, effectively turning RAS off. And so if you lose NF1, you lose this negative regulation of the pathway. There is an abnormal amount of signaling, and there is an increased risk of oncogenesis. And so to speak briefly about AML and about the AMLs that we studied in this particular experiment, AML is the most common form of leukemia affecting adults and is characterized by an abnormally high white count. And to generate the AMLs that we studied, we took a population of mice that were NF1 null in the myeloid system and infected them with an oncogenic retrovirus that caused them to develop AML. And we then took a subset of these AMLs and treated them with a MEK inhibitor drug, PD901, and this prolonged survival, but caused the outgrowth of drug-resistant clones. And these are the two primary populations of mice that I studied this summer, the drug-resistant mice versus the parental clones. And so the primary question that we wanted to ask about these populations was, do NF1 deficient AMLs remain dependent on NF1 loss for leukemic maintenance? And the way that we decided to attempt to answer this question was through an experiment called a colony assay. One of the ways was through a colony assay. And in a colony assay, a set of primary bone marrow cells are harvested from mice with AML. They're then manipulated, and then a known quantity is plated in methylcellulose culture to look for the ability of the population to form cell colonies. And so to show you the most relevant part of that data, we found that both parental and resistant clones do in fact remain dependent on NF1 loss for leukemic maintenance. Just to make this graph more clear, the y-axis is number of colonies per plate, and this population here is a GFP control, a green fluorescent protein control. This population in the middle is a population expressing the gap-related domain of NF1, or the GRD, so we express a truncated form of the protein instead of the entire NF1 protein. And this bar over here on the right is expressing a catalytically inactive isoform of GRD, catalytically inactive isoform of GRD. And so, as you can see in both the 6537 parental population and the 6554 drug-resistant population, with the re-expression of the gap-related domain of NF1, there is a severely impaired colony forming ability when compared to controls. And so our main conclusions from these experiments were that parental NF1 deficient AMLs do in fact require NF1 loss for leukemic maintenance. And we then noticed that in one particular line, the parental 6537 line, the presence of saturating amounts of growth factor were able to rescue this effect. And then in the 6544 resistant line, the clones also remain dependent on NF1 loss, even in the presence of saturating growth factor. And so now I'd just like to take a moment to acknowledge all the people who have helped me be here today. So big thank you to Dr. Michael Burgess for being such an inspiring mentor, to Dr. Kevin Shannon for having faith in a high school student, to everyone in the Shannon and adjacent labs for being so accommodating and so ready to answer questions and help me really at any moment, to Julia and Mr. Shapiro for running an extraordinary program, and to the Lowell Alumni Association for being so generous with funding. Thank you.